Okay, I apologize for getting kind of cut off last time. Somewhat of a family emergency at home. If we were sitting in a classroom right now, I'm sure I would tell you a very amusing story about the home life at the Fagelson household, but we'll have to save that, I suppose, for graduate school. In any event, I think we left off mentioning the four types of structures for which you'll be responsible in with respect to nervous tissue in the peripheral and central nervous system. So we had nerves, which we said were bundles of axons, bundles of axonal material, and again, recall that looked like back up here, right? Axonal material in the peripheral nervous system, so that's a nerve. And then next we skip down here and illustrated, highlighted this collection of cell bodies, sort of like a little swelling. In this case, it's in the inner ear, in the cochlea itself. This brown area here is actually bone, the bone of the skull, and you can see how this swelling kind of sits in the bone and serves almost as a little way station or a point of contact between thousands of nerve fiber bodies, nerve cell bodies. Collections of nerve cell bodies in the peripheral nervous system then are ganglia, which is the plural of ganglion. And again, here we see where it would be located inside the inner ear. As we'll see later in the semester, because the inner ear spirals around itself like a spiral staircase, this is actually called the spiral ganglion. In the central nervous system, the collection of cell bodies would be known as a nucleus. The plural would be nuclei. And you can think of these again as sort of like relay stations or points of contact for thousands, perhaps hundreds of thousands of nerve cells, which will have connections with one another and will contribute to the processing in the auditory system of sound. There are a lot of different nuclei throughout the central nervous system. Of course, for the hearing part of the class, we'll be focusing on those that are associated with the auditory pathway, but there can also be motor nuclei as well. So, for example, there's a facial motor nucleus, which serves as a collection point, if you will, a relay station for those nerves that are going to innervate movement of the facial muscles. That's kind of what's meant by this idea of integrating neural activity, potentially from several sources. So whereas a collection of axons in the peripheral nervous system was known as a nerve, when that same collection of axonal material is found in the central nervous system, it's referred to as a tract. You can kind of think of these as conduits or pipelines that are going to connect different areas of the central nervous system to one another. So let's use the auditory pathway as an example of these structures and kind of see if we can get them all labeled right here. So again, this is Frank Netter's simple schematic diagram of the auditory pathway. Out here would be the actual peripheral end organ of hearing, kind of a slice through the cochlea. Again, much more on that later. Here are individual nerve fibers, these lines right here, exiting the sensory cells, the so-called hair cells of hearing, which are the devices in your inner ear that generate or trigger nerve activity. Now, these nerve fibers are collected together, and you can see for a very short distance right there. And of course, then that would be the sort of the start of the auditory nerve, the so-called eighth cranial nerve. Here we see a collection of cell bodies, kind of like the image we just looked at. So that would be the spiral ganglion. And then the nerve would emerge from the spiral ganglion and make its way into the central nervous system. Its synapses, that is to say, it plugs into or connects to the central nervous system at the ventral or the dorsal cochlear nuclei. So these red areas here, labeled ventral cochlear nucleus or dorsal cochlear nucleus, are collections of cell bodies in the central nervous system. We then see projections of fibers from these nuclei to other nuclei in the auditory pathway. In this case, it's the superior olivary complex. Of course, that would be a decussating, a set of decussating fibers because they cross the midline and are routed contralaterally. But in any event, here's yet another nucleus, the superior olivary complex. And then these two big trunks of nerve fibers are called the lateral lemniscus. If you take them together, it's the lateral lemnisci. And these fibers would ascend. They are essentially afferent pathways that are going to ascend through the central nervous system up to the part of the brain where sound is processed. That would be the primary auditory cortex. And you see that there are a couple of way stations, stopping points, integration points, however you want to look at it along the way. And these are nuclei. So you've got nerve out here, ganglion out here, nuclei all throughout this pathway, and then the tracts in the central nervous system connecting all these different parts together. Ultimately, of course, the signals do reach the brain, and this is actually, this image represents the actual color of the brain. It's a Not only is it a fine integrator of information, it also appears in these wonderful day-glow colors. I am kidding, of course, but these are the different parts of the brain, the different actual lobes of the brain, color-coded here for your viewing pleasure. Notice how we've got this midbrain still sticking up into the base of the brain, emerging from its caudal aspect, right? And again, we had the cerebellum wrapped around in there as well. Now, these different lobes have different jobs, if you will, different functions. And it sure would be nice if we could, in a very fixed and consistent way, indicate function, map function onto location of the brain. We can do that with some degree of success, but it's not as clean as we'd like. And I'm just going to leave it at that for now. But just recognize that the brain, it does consist of several different portions, different lobes, and it is the skull, of course, in which the brain sits. The bones of the skull actually take on or share the same naming as these different lobes. So, for example, you have an occipital bone, which is back here on the posterior section of the brain. You've got a parietal lobe, which kind of is in the middle and covers that dorsal surface of the brain. You've got a frontal bone, which covers the frontal lobe, and the temporal bone, which covers the temporal bone. Another way to think about mapping function to location in the brain is to recognize that to some degree we can see how different body parts are represented by nerve endings, nerve fibers, nerve tracts actually, that can be exposed on the cortex of the brain. Now, this was discovered back in the 50s by a couple of neurologists who did split brain studies on epileptic patients. And one thing that they got permission to do, they were in Canada, and one thing they got permission to do from the Canadian government was to, stick some electrodes and stimulate parts of the brain electrically, just to kind of see what parts of the body were activated when different parts of the brain were electrically stimulated during these surgeries. Kind of a kind of an interesting way to map the brain. And so they found, for example, that if they stuck an electrode right in here to this location, the person's thumb would move up and down or something. The person would be conscious during this, believe it or not. And the investigators, by the way, were Penfield and Roberts. But yeah, you'd stick the brain with an electrode somewhere here, and a part of the body would move, say the thumb, or the person's elbow would bend, or their arm would start waving around, etc., just depending on where that electrode was advanced into the cortex. So we know pretty well that you can map a person's body onto their cortex motorically and with respect to tactile sensation. This little guy's face here and this sort of miniature body would sometimes be referred to as a homunculus, or actually the little man or little person, probably would be more appropriate, that resides mapped out onto the surface of our brains. For those of you who really like to turn back the clock a little bit, there have been other systems for mapping out brain activity, or mapping out human activity onto the surface of the brain. Some of you may remember phrenology or have heard of phrenology, and it's kind of interesting that back in the day there were different parts of the brain and the skull that were to which were attributed many different sort of behavioral or psychological aspects. So, for example, if somebody had a nice lump on their skull right up in this area here, it might be determined that they had a lot of perseverance, that they could stick with something, had a great work ethic, for example, whereas lumps a little closer to the front of the skull seem to be associated with people who were very kind. And you can see there are different kinds of things here, like cautiousness and love of friends and love of life, etc. We kind of laugh at this now, but who's to say that in 10,000 years or so, people won't be laughing at this just as much? Here are some of the specific areas in the brain kind of separated out from the lobes in which they would otherwise appear. And for our work, Broca's area and Wernicke's area for speech will, as you'll see, be real important. And for the hearing folks, this primary auditory cortex is important. Notice how these areas, even though they include motoric and sensory areas, are located right next to each other. We would recognize from that there would probably be some connections, neural connections between these areas that would foster the functions contained therein. So we would expect then that hearing should have an influence on word recognition and maybe even word production because these areas are so close to one another. Just kind of reinforces the idea that audiologists and speech-language pathologists actually do have a lot of information to share with one another with respect to the treatment or the diagnosis of patients. I tossed this in just to give a little idea of how different kinds of imaging studies may represent different functions within the brain. Can anybody tell me what kind of slice through the head this would be? I'll give you a hint. The nose would be sticking out like this, and it's a view from the top. Of course, it's not the dorsal surface of the brain, but it's deep to that. So this would be a transverse slice through the brain at some level. This would be the left side of the person. This would be the right side of the person. And again, the nose, the front, the anterior surface would be that way. The different colors just represent the amount of nerve activity, or I'm sorry, brain activity going on at any given moment when the individual is stimulated. And by comparing the amount of normal activity, the amount of activity in a normal brain to the amount of activity in a brain of a person who has Parkinson's disease, you can see this different coloration here indicates there's different levels of activity. The redder the area, the more activity there is when the person is completing a specific task. And so here in the normal brain, you see a lot of activity going on with this specific task, whereas in the brain of the person who is suffering from Parkinson's disease, that's not necessarily the case. However, maybe we give them some medicine, some training, some help, and look, the brain seems to become more active post-treatment. So these imaging studies are really interesting, kind of beyond the scope of this class, but I thought I would toss it in there just to give you an indication of some of that work. I think this speaks for itself. We would have the brainstem, Homer's brainstem, of course, right there. It's interesting that Homer's brainstem actually, to me, appears to be disproportionately large relative to the cortex. That's why Homer can still breathe. He can still receive sensory information, autonomic activity, heartbeat, all that stuff. He's still alive. He just may not be the most, thoughtful or considerate person from time to time. So the body systems that we've looked at, we're going to be recognizing that they are all part of a whole, and we'll be focusing in, of course, on those body systems that are, or those elements of the body systems that are associated with speech and hearing. Specifically, we'll be looking at the respiratory system, phonatory, articulatory, and auditory systems, and it will be the anatomy and physiology of these systems that form the meat of this course. So in sum, you can just kind of read through this slide, and if you have any questions, definitely check that discussion board. I just started looking at it myself, answered one question on it, and we'll be sending out those quizzes in a timely manner. Again, discussion board is one way to get hold of us. We'll be also trying to offer some sort of study group things, and we're going to try to record those for people who can't make it to them. And of course, we have office hours as well. So don't be a stranger. Come on in anytime you need to, and make sure that you do not get too far behind with respect to this material, because the momentum of the course will build pretty quickly. Thanks a bunch. Hope to see you soon.